Hello everyone, and welcome to this lesson on the structure and function of membranes. To start, let's remind ourselves that we're all made up of tiny little structures called cells. The cell is the basic unit of life, and as such, they do a lot. These little structures are constantly carrying out a whole bunch of processes that are absolutely essential for life. In order to do that, the cell needs to be in constant contact with its surrounding environment. For example, it takes raw materials into itself from the surroundings and removes waste products by sending them into the surroundings. In order to do that, the membrane needs to be in contact with the surroundings and all the substances that may be floating around there. In case any substance there wants to enter the cell, the first thing that substance encounters is the cell membrane, and it's the cell membrane that determines whether or not that substance will be able to get through. It's kind of like a bouncer that lets some people into the club but turns others away. In fancy science terms, that's called being partially permeable, meaning that it lets some things through but not others. Now, even though we're going to be talking about the cell surface membrane in this lesson, membranes that surround organelles, such as the nucleus or mitochondria, also have pretty much the same basic characteristics. So remember, while we're talking about the cell membrane here, most of this will also hold true for, let's say, the nuclear envelope or the mitochondrial membranes. The cell membrane, chemically, is made up of these little structures called phospholipids. They look like little octopuses, except instead of 8 legs, they have 2 tails. This part is called the head of the phospholipid, and this part is called the tail. These phospholipids arrange themselves into 2 layers, with the tails facing each other and the heads facing both the inside and the outside of the cell. This is called a bilayer, which means 2 layers. We'll talk about why the phospholipids arrange themselves in this way in a bit, but for now, understand that the cell membrane is made up of a phospholipid bilayer. Now, here's where it can get a little confusing, so let's understand this properly. While the phospholipids are arranged in 2 layers, when you look at the cell membrane under an electron microscope, it looks like there are 3 layers. These 3 lines you see here are actually a single membrane under the microscope. The phosphate heads stain more darkly, and the tails of the phospholipids appear as a different color when you stain them, since they take up less stain. And so all the tails look like one single layer sandwiched in between 2 layers of heads. This gives the appearance of having 3 layers, and it's called a trilaminar appearance. The word tri means 3, so this means 3 layers. Remember, it's not actually 3 layers, it's actually a bilayer, but it has a trilaminar appearance under the electron microscope. Okay, so let's now look at the structure of a phospholipid. This will help us understand why they arrange themselves as a bilayer in the first place. The phospholipid, as the name suggests, is a lipid, but it's a lipid that has been modified by the attachment of a phosphate group. The lipid part of the molecule is found in these fatty acid tails, so you'll sometimes see people refer to the tails as fatty acid tails. The phosphate head, because of its phosphate group, is negatively charged. Now, being negatively charged makes the phosphate head polar, which means that it can dissolve in other polar substances. This is why the head is referred to as being hydrophilic, which means water-loving, since it readily dissolves in water and other polar substances. That means that these heads will be able to dissolve in both cytoplasm in the intracellular space and tissue fluid in the extracellular space, both of which are aqueous environments. The tails, on the other hand, are nonpolar. This makes them hydrophobic or water-fearing, and they do not dissolve in polar substances. With the heads of the phospholipid being hydrophilic and their tails being hydrophobic, the phospholipids naturally arrange themselves with the heads facing both the internal environment of the cell as well as its external environment, which are both polar, and the tails remain protected in between them.
Quick pause. If you're finding value in this explanation, there's so much more to discover at Alt Academy, where we break down every A-level concept with the same clarity. Now back to the video lesson.
Since the phospholipids that make up the cell membrane are in fact lipids, they're going to have a similar consistency to some other lipid substances, such as fats that are solid at room temperature or oil that is liquid at room temperature. So the question becomes, which of these consistencies is our cell membrane more similar to? In our body's environment, our cell membrane tends to be closer to the olive oil in consistency. And so, just like olive oil, it too has the ability to become more fluid or freeze up to become solid depending on the conditions it's in. But if it were solid, then that would block the exchange of substances. So cells work hard to maintain the olive oil-like consistency, so to speak. So our cell membranes are not solid structures and actually remain quite fluid in nature. Alright, now that we've understood the structure of these phospholipids, let's see what happens when substances surrounding the membrane try to pass through. Now, small non-polar substances like oxygen and carbon dioxide can simply diffuse across the membrane because they won't be repelled by the non-polar tails. However, when it comes to polar substances, such as these ions, this is not the case. These substances are repelled by the hydrophobic tails, so they can't simply diffuse across them. And that's a big problem. Many of the substances that the cell needs, like these ions here, are polar, and so are a number of waste products that the cell needs to remove. So if the cell is to survive and carry out its functions, it has to have a way to transport the polar substances it requires into the cells and those that it needs to remove out. As with most things in the body, the answer to this problem is proteins. You see, the cell membrane isn't just made up of phospholipids. It's also studded with many proteins, and it's the role of these proteins to allow some of these polar molecules to pass through the membrane. Notice how I said some, which means that these proteins will only allow the entry of some polar molecules while restricting the rest. What substances will be allowed entirely depends on what protein is present in the membrane. For example, a protein that allows entry of glucose, let's say, will not allow entry of the chloride ion, and vice versa. Now we can understand what it means when we say that the cell membrane is partially permeable. The nonpolar tails restrict the movement of polar substances, and depending on which proteins the membrane has embedded within it, only some of those polar molecules can get through. In addition to this, if a molecule is too large for the proteins or to fit through the spaces between the phospholipids, then they also will be unable to pass through the membrane. The cell surface membrane doesn't just serve as a partially permeable barrier between the cell's internal and external environments. It also helps the cell attach itself to other cells around it to form tissue, and it even plays a role in helping the cell to communicate with other cells that are far away. In order to do all this, it makes use of structures called glycoproteins and glycolipids. If you attach a carbohydrate chain onto one of these proteins embedded in the membrane, that's called a glycoprotein. And if you attach it to a lipid, that's called a glycolipid. These glycoproteins and glycolipids can be different on different types of cells and behave as markers or antigens for the cell, which can be used by other cells and molecules to identify them as them. They also use these markers to communicate with each other from far away by sending molecules that function as messengers to bind to these specific markers, which serve as receptors or binding sites for them. So, in this way, these glycoproteins and glycolipids allow the cells to send signals to each other, even if they're in different parts of the body. These kinds of functions are only possible for the cell because of how the proteins are embedded in the cell membrane. Some of these proteins are only on the surface of one of the layers of the membrane, either the outer layer or the inner layer. These are called peripheral proteins. Other proteins will run through the entire membrane, all the way from one side to the other. These are called transmembrane proteins, and they connect the cell's internal environment to the external environment. These proteins appear as little black dots when viewed under a microscope after using something called freeze fracture or freeze etching. Freeze fracture is a technique that physically breaks apart or fractures a frozen biological sample to be seen under an electron microscope. When this technique is applied to a membrane, it causes the bilayer to be separated into two separate layers. The outer layer is called the E face, and the inner layer is called the P face. And a dark line of fracture can be seen in the center where they separate. You can imagine this as being like a book with a bookmark in the center. The upper half of the book is the upper layer, and the lower half is the lower layer. Freeze etching is basically opening that book, separating the two layers from where the bookmark is, and that line along which you open the book becomes your line of fracture. Under the microscope, it looks like a dark line, as you can see here. This is where the two layers were separated. Usually, the P face is studded with more proteins than the E face, so it looks a bit rougher, and the E face looks a bit smoother. What this tells you is that the inner layer of the cell membrane has more proteins than the outer layer. Okay, so we've established how the cell membrane is a phospholipid bilayer with a fluid-like consistency. This means that the phospholipid molecules are not fixed in place, and they are constantly moving around. So let's create a model to understand this better. What I want you to do is imagine a deep dish that has water in it, okay? The water will represent the cytoplasm of the cell. Now, add some yellow beads in your dish, all right? Imagine this. You will assume that these yellow beads are your phospholipid molecules. If you observe these yellow beads, you'll notice that they're not going to be still. They will constantly be moving from the areas that there's more beads to the areas where there's more space. The phospholipids in our cell membrane diffuse and move about in a similar way. We also talked about how the membrane has many proteins embedded in it. So now, if you add some colored beads or balls representing these proteins, you'd notice that they're also moving like that. Looking at all these different components from the top of this deep dish, scientists decided to call this the fluid mosaic model. The word fluid refers to the fact that the phospholipids are able to dynamically move across the membrane surface, and the word mosaic refers to how the whole structure looks when viewed from above, because of the scattered arrangement of proteins in between the phospholipids. If you've ever seen a mosaic, it's actually made of lots of different little parts that are placed together to form one complete picture. But if you look at it closely, each of the little parts is different from the others. In a similar way, all the proteins in the cell surface membrane can also be different from each other, and they're scattered between all the phospholipids. Generally, the word mosaic refers to a design that's made using small irregular pieces of glass or stone, and in the model of a cell surface membrane, the scattered arrangement of proteins between the phospholipid makes this resemble a mosaic, and hence the term mosaic in fluid mosaic model. But keep in mind that despite the cell surface membrane being such a complex structure, it's very thin, right? The cell surface membrane is only about 7 nanometers thick, and yet it's able to carry out all of its functions and hold all the cell's contents within it, making it a really incredible structure. Finally, let's summarize the lesson. The cell surface membrane is a partially permeable structure made up of a phospholipid bilayer with a trilaminar appearance under an electron microscope. It consists of membrane proteins that allow the controlled entry of polar molecules that would otherwise not be able to pass through. We also talked about glycoproteins and glycolipids and how they can help the cells in cell-to-cell adhesion, cell recognition, and cell signaling. Finally, we went over the fluid mosaic model and saw how the fluid nature of phospholipids and the scattered arrangement of the proteins led to that name. And with that, you should now have developed a general idea of what a membrane is. I hope you found this lesson helpful, and I'll see you in the next one.
Hey there! If you found this A-level biology lesson helpful, why don't you head over to altacademy.org and sign up for our full A-level biology course. In addition to biology, you could sign up for physics, chemistry, math, or 4 other A-level subjects. Thousands of students have already taken the leap. What are you waiting for? See you there.